Ever wondered how our body fights against foreign substances? It's a marvel of microscopic warfare, a complex dance of recognition and response. This battle is defined by antigen-antibody reactions, a crucial component of our immune system. These reactions act as our body's personal security team, identifying intruders and responding accordingly. It's a world of precipitations, agglutinations, and an array of fascinating tests. So, fasten your seatbelts and prepare for a journey into the microscopic battlefield within us. Stay with us as we delve into the fascinating world of antigen-antibody reactions. So, what exactly is an antigen-antibody reaction? Well, it's a fascinating dance of molecules that happens within our bodies every day, and it's essential to our survival. In its simplest form, an antigen-antibody reaction is a biological response where antibodies, produced by our immune system, recognize and bind to antigens, which are harmful substances like bacteria, viruses, or toxins. Now, let's delve into the key characteristics of these reactions. First up is specificity. Each antibody is specific to a particular antigen, much like a lock and key. This specificity is determined by the unique structure of the antigen, allowing the antibody to recognize and bind to it. Next, we have reversibility. Antigen-antibody reactions are not permanent. They're reversible, meaning the antibody can detach from the antigen. This is crucial in the dynamics of our immune response, allowing flexibility and adaptability. Last, but certainly not least, these reactions are observable. We can literally see them occur in laboratory settings. Through tests like precipitation, agglutination, immunofluorescence, and more, we can visualize these reactions, aiding in the diagnosis of various diseases. So there you have it. Antigen-antibody reactions are specific, reversible, and observable. They're a testament to the complexity and adaptability of our immune system. Understanding these characteristics is the first step in comprehending the complexity of our immune response. Now, let's move on to the basics of precipitation. In the realm of immunology, precipitation isn't about rain clouds, but rather the interaction between antigens and antibodies. This process is a crucial part of identifying and understanding the presence of specific antigens in a solution. Imagine this. You're in a room filled with people. You're looking for your friend, but it's challenging because everyone is moving around. Suddenly, everyone freezes. You can now easily spot your friend. This is similar to what happens in a precipitation reaction. The antigens, or friends, are moving freely in a solution. When the right antibodies or freezers are added, they bind to the antigens, forming large complexes that settle out of the solution or freeze. This process is a type of immune response, where the antibody, produced by the immune system, recognizes and binds to the antigen, which could be anything foreign to the body, like bacteria or viruses. The resulting antigen-antibody complex is visible to the naked eye, making it a useful tool in laboratory diagnostics. This isn't just a random meeting, though. The interaction between the antigen and antibody is specific and highly selective, like a lock and key. The antibody can only bind to its corresponding antigen, ensuring that the immune system targets the right invader. Precipitation plays a vital role in the visibility of this reaction. Next up is agglutination, a process you might not have heard of, but it's crucial. Now, what is agglutination? It's a reaction that occurs when an antigen is mixed with its corresponding antibody, known as agglutinin. The result? Clusters or clumps, which are easier to see and identify. Agglutination is critical in many laboratory tests used to diagnose various diseases, and it's also important in blood typing. So let's delve a bit deeper into the slide and tube methods of agglutination, shall we? Imagine a simple slide test. A drop of serum is mixed with a drop of antigen on a slide. If agglutination occurs, it indicates the presence of the corresponding antibody in the serum. It's a quick, straightforward method often used for blood typing. Then there's the tube method, a more sensitive and quantitative approach. Here, serum and antigen are mixed in a test tube. The tube is then incubated and observed for clumping. The degree of agglutination can tell us about the concentration of antibodies, making it a valuable tool in diagnostics. But agglutination isn't just about clumping. Its value lies in its ability to make tiny, almost invisible entities visible to the naked eye, allowing us to detect and quantify them. It's like a magnifying glass for our immune response. Agglutination, though a complex process, is a cornerstone of antigen-antibody reactions. It's a fascinating world, isn't it? And we're only just scratching the surface. Now, onto some more advanced concepts: complement fixation, immunofluorescence, radioimmunoassay, and ELISA. Complement fixation is a fascinating technique that takes advantage of the body's natural defense mechanism. It's like a biological detective looking for evidence of an immune response. Complement proteins are part of our immune system, and they can bind to antibodies that have attached themselves to harmful invaders. In a complement fixation test, we use this to our advantage. If the complement proteins fix themselves to the antibodies in a sample, it's a clear sign that an immune response is underway. This test is particularly useful for diagnosing infections that can be harder to detect, like certain bacterial and fungal infections. Next up is immunofluorescence, a technique that brings a touch of color to our exploration of the immune system. In this process, we tag antibodies with fluorescent dyes and then let them interact with a sample. When we look at the sample under a microscope, the glowing antibodies reveal where and how they've latched onto antigens. This technique is invaluable in research and diagnostics, helping us visualize how our immune system interacts with pathogens. Now let's talk about radioimmunoassay, or RIA. This technique sounds like it's straight out of a science fiction movie, but it's actually a cornerstone of modern immunology. In RIA, we use radioactive isotopes to label antibodies or antigens. When these radioactive labels bind to their targets, they emit radiation that can be measured. This allows us to quantify minute amounts of substances like hormones or drugs in a sample. The precision and sensitivity of RIA have made it a crucial tool in many areas of medicine and research. Last but certainly not least, we have the enzyme-linked immunosorbent assay, better known as ELISA. This technique is a bit like a high-tech game of tag. We attach enzymes to antibodies, then let them bind to antigens in a sample. When we add a special substrate, the enzymes cause a color change that can be measured. The intensity of the color change tells us how much of a particular antigen is present. ELISA is widely used in diagnostics and research, and it's particularly effective for detecting viruses like HIV. Each of these techniques, complement fixation, immunofluorescence, radioimmunoassay, and ELISA, provides us with a different lens through which to view the complex dance of antigen-antibody reactions. They've expanded our understanding of the immune system, shedding light on how it recognizes and responds to an immense variety of foreign substances. These advanced techniques have revolutionized our understanding of the immune system and how it fights off foreign substances. So what have we learned today? We've journeyed through the fascinating world of antigen-antibody reactions, unveiling how these microscopic interactions form the bedrock of our immune system. We've dissected the characteristics of these reactions, understanding how specificity, avidity, and cross-reactivity shape our body's response to foreign invaders. We've delved into the basics of precipitation, a process where soluble antigens and antibodies come together to form large, visible aggregates. It's like a microscopic tango dance, where the partners are so perfectly matched, they become inseparable, precipitating out of the solution. Next, we navigated the realms of agglutination, both on a slide and inside a tube. Agglutination, a process where particular antigens clump together in the presence of specific antibodies, is a technique widely used in blood typing and diagnosing infectious diseases. It's like a tiny traffic jam caused by our body's defense mechanism. Then we ventured into the advanced territory of complement fixation tests, immunofluorescence tests, radioimmunoassay, and ELISA. These techniques are like detectives, helping us uncover the presence of specific antigens or antibodies in our body. Complement fixation tests and immunofluorescence tests provide a snapshot of our body's immune response, while radioimmunoassay and ELISA, with their high sensitivity and specificity, are the gold standards in diagnostic immunology. From characteristics to advanced techniques, antigen-antibody reactions are a testament to the marvel that is our immune system. They illustrate the intricate, delicate balance that keeps us healthy, and the sophisticated tools that allow us to unravel these complex interactions. As we continue to probe the mysteries of our immune system, we gain not only a deeper understanding of our bodies, but also the potential to develop more effective treatments and vaccines. Remember, the field of immunology is vast and ever-evolving, and there's always more to learn. So keep that curiosity alive and continue to explore. From characteristics to advanced techniques, antigen-antibody reactions are a testament to the marvel that is our immune system. Subscribe for more because you won't find me later.